This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 432, recorded on March 10th, 2017. This episode of TWiV is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with your first purchase with free shipping by going to blueapron.com/TWiV. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me here today in a cold but sunny New York City, Dickson Despommier.
Hello, Vincent.
How are you?
I'm fine. By the way, you forgot to mention that it was snowing like crazy in the morning.
Well, this morning it was.
Yeah.
I don't give the whole week's weather in one shot. You know that.
But today, at least.
Yeah, it snowed, but just a few inches, right?
Yeah, not even that. I mean, it's almost all gone off the ground now.
2 degrees Celsius.
Yeah.
Also joining us from, what?
Sorry, I was going to ask, what was it yesterday?
I don't know.
It was probably over 60.
No, not that warm.
It was pretty warm
but
yesterday.
But it did dramatically drop, yeah.
It did.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's the snow here cleared out maybe a couple hours ago, except for a few flurries, and it's and the wind has moderated at least temporarily. But so it's what is it now? It's out of the north. It's 7 knots, not too bad, and temperature's right at freezing.
Do you have any accumulation?
It was odd. It accumulated on the grass, but not on the roads or the sidewalks. So
right.
It was just very, very pretty. It's fine with me.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's 28 degrees Fahrenheit, -1 Celsius. And remember, Vincent, last week we talked about the squiggly lines on your app?
Yeah.
I don't know how bad the winds were in New York, but they supposedly got up to 60 miles an hour here. Wow. All kinds of power outages. We had power out for 75 minutes in part of the building, but not the end of the hall where Alice's lab is. It was we almost had to cancel my class. Sticks and stuff down all over. There's some people around the area who won't get power back until next week. Oof. It's really bad.
I heard the Detroit area really got slammed.
Yeah.
As well. Yeah.
Yeah. Well, that's,
Wow.
We're only,
Not too far.
30 minutes from the Detroit airport. Yeah. So, yeah.
We had some wind this week, right?
Yeah, we did.
I don't remember when.
We did, yeah.
Actually,
Wednesday.
But that was a convention of politicians. That's what that.
That's it.
We have an in-studio guest today. She is a professor of biology at Drew University. Brianne Barker, welcome to TWiV.
Hi, thank you.
In the TWiV studio in Columbia University. And all that. You're a TWiV listener, right?
I am a TWiV listener.
We met, Alan and I met you last summer in Boston at ASM, right?
Yes. My colleague, Joanna, 2 students, and I were there.
So Joanne is also at Drew?
She is.
And was that what we were just talking about, Kathy? Joanna? Or someone, maybe a friend of Joanna knew Kathy? Okay, got it.
Knew one of my former students.
Yes.
Got it. So you're on spring break this week.
This is the end of spring break.
Back to work next
And so
week.
where did you go for your spring break? Well, I went to the TWiV studios.
Hey, don't make fun.
Really?
For the beginning of spring break, I went to my own lab and did some pipetting. And then at the end, I came to the TWiV studio.
Exactly.
So tell us your history. You're from upstate New York, right?
I'm from a very small town in upstate New York called Camden, which is near Utica and Rome. Then I did my undergraduate degree at Duke. I got a PhD in immunology at Harvard with Norm Letvin, and did a postdoc at UNC Chapel Hill. And now I have my own lab at Drew University, where I work with undergraduates in my lab and teach a bunch of different courses.
What did you do your postdoc in? What area?
It was in immunology and understanding innate immune receptors.
With who?
Jenny Ting.
Mm-hmm. Sure. So did you want to work at a relatively small undergraduate university or college, right? Drew University is a university, so it's got several colleges within it. Right?
It is. We only have undergraduate programs in the sciences, so it's an undergraduate institution. I was on the fence as a graduate student, although I really enjoyed teaching.
Yeah.
And when this opportunity became available, I realized how much I enjoyed mentoring and teaching in graduate school.
So you've been there how long now?
I'm in my 4th year.
Great. Well, and your research involves innate immunity, right?
Yes, I'm working on understanding innate immune receptors that respond to viral nucleic acid, as well as how those receptors are evolving in a few different primate species.
And you primarily use undergraduates in your research? Not use them. Doesn't sound right.
It's cheaper than a mouse facility.
Well, we do have a mouse facility. We actually are working with cell lines, and my lab is entirely undergraduate students and myself doing all the work. So I have 6 students right now,
Cool.
And myself doing all of our experiments.
So you're in there on a daily basis doing experiments?
I am.
You think that'll last forever?
Probably. I really enjoy it.
You like to do that? Yeah.
So I have my schedule set up now so that I teach in the mornings and then have the afternoons for experiments.
Nice. Dixon's friend is out there at Drew. Well, maybe twice a year he's out there, right?
She knows him, too.
I do.
Bill.
And who would that be, Dickson?
That would be William Campbell.
Yeah. Is that your only Nobel laureate?
It is Drew's only Nobel laureate. We are lucky enough to have a research institute for scientists who have retired from other positions in the top floor of our science building. Those folks get some lab space and are able to work with students in the lab.
That's great.
That's amazing.
That's just great. Just like we do here, huh, Dickson?
Absolutely. We wish.
Well, I mean, you do have space, Dickson.
I'm not complaining.
You don't have a lab, though.
I'm not complaining.
But there are no undergrads here, right?
There aren't. So that's why I go down to Fordham to teach, because
Yeah.
that's where the undergraduates are.
This is good. Do any of your students know about TWiV?
They do.
Okay.
I have a few students who listen quite intently.
So they'll be excited to hear you on, right?
They will.
All right, and then this will get you tenured, no doubt.
I hope so.
Depends on what you say.
What, does anyone listen?
Absolutely.
It's just the appearance.
You read these letters, everybody's listening.
Yeah, that's true. They are listening. I want to tell you about 2 meetings. First, NIDO 2017, Ying Fang and Susan Baker are organizing the 14th International Nidovirus Symposium. What a neat name, Nidovirus.
And if you play Pokemon Go, you could probably catch a bunch of Nidoron there.
Oof.
Yep. 4th through 9th of June in Kansas City. I'll be there. Be doing a TWiV from the meeting. Unfortunately, the early bird deadline has passed, but online registration is still open. There's a symposium overview online, which we'll link to. You can see who's going to be speaking there. This is an open international meeting, as the meeting organizers recognize that the global community benefits from collaboration and sharing of ideas. I hope that wasn't a political statement. You know,
I can't imagine why they felt the need to even include that.
No, not at all. By the way, they don't just have Nido viruses there. Although all viruses are Nido, they have other viruses be represented as well. It's quite a nice meeting. Also want to tell you about the ASM Conference for Undergraduate Educators. That would be you, right?
Yes, that would be me.
Do you know about this conference?
I do. The Q conference is great.
Yeah, ASMQ. Premier event in the field designed specifically for microbiology and biology educators. And in 2017, it's going to be in Denver, Colorado, July 27th through 30th. And you should go to this meeting to participate in formal and informal conversations that will enhance the work you do every day and shape the future of teaching and learning in the biological science. Secure your seat today and save money, and submit a microbrew oral abstract by March 15th. I have no idea what a microbrew oral abstract is, but presumably if you go to asm.org/ASMQ, you can find out. And you have till April 3rd to submit a poster abstract, which is not too far away, so you should get moving.
I presume those abstracts will be beer reviewed.
Dickson, you were on a roll today. Amazing. I went to this ASMQ when it was in Boston, I think 2 or 3 years ago.
Yes.
Did you go to that one?
I did. I always get a lot of useful information that helps my teaching at the Q meetings.
So I gave a talk about viruses in general, but also resources that would be useful for undergraduate educators. It was lovely because people came up to talk afterwards, and they had to throw me out of the room because there was another session coming up. And but I really enjoyed it. I got to meet a lot of people that I wouldn't otherwise. All right, we have a few follow-up emails. The first few have to do with politics on TWiV. Joel writes, seconding the vote on dumping politics as a continuous subject on TWiV. How about if you bring it up again when you have a genuinely new idea on it and not the same old tired complaints? Act as if I'm as bright as you. I get it the first time. Okay. I respect everyone's views, but you don't have to be nasty. I think that's a nasty statement. We talk about politics in the context of science, not other kinds of politics. And this is the appropriate forum from that. I don't think we have the same old tired complaints. You know, if the FDA is going to be changed, we're going to talk about it. If NIH budgets are going to be cut, we're going to talk about it. And we do respect all of our listeners. We think they're just as bright as everyone else. So I think that's an unfair comment. Anthony writes, should scientists be involved in politics? Today, it is no longer the cholera or plague bacillus that threatens us, but the traditional, cynical reasoning of politicians, the indifference of the masses, and the physicists and other scientists' evasion of responsibility. That's a quote from Max Born, and he gives a link to that. And he, at the end, he writes...
From 1964.
Thank you. Yeah, this has been going on a long time. He writes, scientists don't have the right, they have the obligation.
There it is.
Kathy, could you take the next one?
Sure. Adam writes, greetings, leaders of the TWiVites. I'm writing in strong disagreement to Hannah from Boulder's email on TWiV 431 regarding the discussion of politics on TWiV. I enjoy the great balance that TWiV provides between both cool virology and the surrounding non-technical aspects that affect virology. Many great benefits come from hearing you talk about subjects falling in the latter category. For example, TWiV 385, Failure, is among my favorite episodes. I'm a senior undergraduate student who has tailored my undergraduate experience primarily to pursuing a career in research. However, years of aggressive funding cuts and other deleterious policies affecting scientific research in my home state of Kansas drove me to become involved in writing and influencing public policy on the state level while still a student. I believe that we as young scientists cannot remain disengaged with this reality, and this especially includes those of us pursuing research careers. There was an excellent interview in ASM's career newsletter written by Adriana Bankston, see link below, about Dr. Kate Stoll, who is the senior policy advisor for the MIT Washington office. Fellow students, many of whom likely share my discouragement, yet strong call to action in light of current political events, may find it interesting and helpful if you had another science policy expert on the show who could talk about opportunities for engagement, specifically in the context of the current situation. It's currently 46 degrees Fahrenheit and clear skies after a day scattered with heavy winds, golf ball-sized hail, a confirmed tornado, and what would have been in previous decades considered to be an uncharacteristic high of 79 degrees Fahrenheit in Manhattan, Kansas. Parentheses, fondly known as the Little Apple. Get it? Manhattan, Kansas. Yes.
Yep.
Okay.
That sounds, other than the high temperature, that sounds kind of like what we associate with Kansas, right? You got a twister and heavy winds.
Yep.
Very true.
Yeah.
I was there a couple of years ago. I did a TWiV there. Adam, you weren't there at the time, but you should check it out.
Alan, I totally agree with you.
What? Oh, Toto? All right, Dickson. You're getting out of hand now. Should we look into Dr. Kate Stoll as a guest on TWiV? Yeah, I'll check it out.
Sure.
Yeah.
Alan.
Yeah. Anyway, it's a nice blog post at the ASM site that he gives a link to.
Right. Alan.
Sure. Trudy writes, Dear TWiVers, I apologize in advance as I'm going to be ranting a little in this email. However, I'm sick and tired of people complaining about the politics on TWiV, at work, in social circles, and on social media. The truth is that we live in a political world. Everything we do is affected by politics. Every privilege that you enjoy today in this free country you live in has been brought about by some political movement or other. The moment you force scientists to ask the government for funding in order to do their work, you have politicized science. Accordingly, if something happens in the political world which threatens or diminishes that financial supply, scientists will and should complain. Why? Because we live in a free country where we're allowed to practice freedom of speech. You do not want to know what it's like to not have that right. I grew up in such a country, and it really made me appreciate true freedom. We must fight to protect that freedom, so if you don't like what you see or hear, then don't look and don't listen. I've certainly had to drive past many anti-abortion rallies by people holding up grotesque photographs and had to remind myself of the same thing. No progress of real value in the history of this planet has been effected by people who were quiet or even politically correct. All progress in every facet of life has occurred due to the acts of people who were courageous enough to insist on change in spite of the consequences. In some cases, these consequences were imprisonment or even death. Here are a few people who said or did things that at the time were considered extremely controversial, but in retrospect literally changed the world. Rosa Parks, Martin Luther King Jr., Sojourner Truth, Charles Darwin, Galileo, Abraham Lincoln, and Harvey Milk, just to name a few. My point is that if you believe passionately in a cause, you should talk about it, no matter how controversial it is or how many people you might offend. That being said, to all the people who are expressing their disgust, fears, and disappointment about our new administration, you have my support. And if you're marching in Atlanta on April 22nd, please come find me. I'll be the one in the TWiV shirt. Ouch. She thinks she'll be the only one in a TWiV shirt in Atlanta?
Be the one, yes.
The one.
That'd be one of you in Atlanta. Wear your TWiV shirt.
Wear your TWiV shirt, yes.
Yeah. Dickson, you want a TWiV?
Sure.
Maybe you wear a TWiP shirt.
I'll
right?
wear a TWiV. I'll wear two, and I'll cut them out, and I'll make half and half. Put the back in the TWiV.
You don't have to. TWiP is your thing. I get it. Go ahead for the next one, Dickson.
Margaret writes, "Hello, TWiVers. Since we're all happy in our pedantry, I'd point out that Orzo, as discussed in relation to Blue Apron ad in episode 431, is not rice. It is a rice-shaped pasta. I'm really sort of shocked that none of the five of you knew this.
Particularly Dr. Racaniello.
Did I say it was rice?
We all said it was rice.
I think I misheard it, but I was only half listening. But I knew it wasn't rice, but I thought you said rice-like or something. But
I might have said rice, yeah. But I know, of course, I know it is orzo.
We mistook.
I make mistakes, and I admit them.
We all do. Well,
we
that's
we
why
don't
we're
always
reading this
correct
letter.
everything, right?
Here are two other podcasts that I listen to showing my nerdiness in other arenas besides science. Pod Save America, politics, current events, lots of snark, and a good bit of swearing from several of President Obama's speechwriters. That should be interesting. The Sweet George's Show, fine arts, especially knitting and color.
Why don't you save the rest for our...
I will.
So the next paragraph is a pick. So go down to thank you, Dickson, please.
Thank you for your great podcast, and I'm bummed that the ant one has fallen through. They make my 2-hour commute each way between Tacoma and Seattle much more tolerable and keep my brain engaged while I knit on the bus. Currently, it's 40 degrees C and raining somewhere along that route.
It was supposed to be a superscript of
I see the other way.
4 degrees
40.
C.
Oh, 4 degrees C.
4 degrees C.
Oh, you're right.
I don't think it gets to 40 in Seattle.
No, 4. That's right. Of course not.
It would be warm.
It would be
Right.
overly warm.
Very well.
Anyway, she really multitasks. She's riding the bus, she's knitting, and she's listening to podcasts.
Yeah.
It's great.
I think we should do an orzo podcast. What do you think? All about
They
Italian
look a little
rice.
they look a little like ants, white ants.
Yes, I'm embarrassed at getting Italian things wrong, but it happens, right? Nobody's perfect. Next one is from Ted Deal, who we had on TWiV some time ago and talked about the Ebola virus mutations that and their effects on infection. I just listened to your most recent TWiV featuring the paper from Jim Cunningham's lab. I wanted to clarify a couple of points that were discussed during the show. One, you referred to the assays done in figures 2A, 3A, and 3C as binding assays, but truly those assays measure GP triggering, exposure of the fusion loop and insertion of this into membranes. True binding assays in the form of ELISAs were performed in figure 4A. That assay showed absolutely no difference between the ancestral glycoprotein and A82V. Yes, I think we did mention that these are measuring the insertion of the fusion loop. Okay, so I don't think we called them binding assays. And certainly the ones they did later, the ELISAs were binding assays. Although you do need binding first to get triggering of the fusion loop movement.
If we did refer to them as binding assays, I think we understood that they weren't true binding assays.
Actually,
you could go back and listen.
I think
we could. Yes,
I think Dickson referred to them as orzo assays.
I did.
Yes.
Right. Number 2, in regard to our paper and the associated TWiV episode, you said that we found evidence for A82V mutation resulting in enhanced binding to NPC1. In our paper, we presented no data either way as to interactions, GP, NPC1. And in fact, we heard of the data presented by the Cunningham group as figure 4A. Rather, in the discussion of our paper, we explored both the possibility that the mutation enhances NPC1 binding as well as the possibility that it increases triggering. Additionally, toward the end of our TWiV conversation, Rich Condit asked us about performing such a binding assay. After my circumspect response, Jeremy chimed in and shared that our current belief was that A82V resulted in a glycoprotein that was more predisposed to conformational changes, resulting in exposure of the fusion peptide. Indeed, this is exactly what the Cunningham report found. So what I was referring to when I said that they did binding assays was here, and in this case, I did go to the original paper. So the Cunningham paper in the discussion, quote, our studies do not support the suggestion that changes in residue 82 alter receptor affinity, and they cite the Deal-Luban paper. That's all I got that from, and I didn't go to check the Deal paper to see that was just in the discussion. And so there you go, orzo and rice. Kathy, last one.
Sure. James Cunningham writes, thank you very much for choosing our paper for your podcast and for providing such a thorough and positive discussion. All of us, but particularly the graduate students, Mei Wang and Sumi Lee, who carried out the studies, were very flattered. They've been bombarded with congratulatory texts, emails from their peers in the virology program, which is the ultimate form of validation for young scientists. So thanks again. It means a lot. And I hope we can discuss some of the details in the future. Best regards, Jim.
Oh, that's nice.
Yeah, I think it's pretty cool that your paper was done. You know, we cannot do every paper. We do, 100 papers a year on TWiV?
Yeah.
If that?
Mm-hmm.
Or
like so.
Or so. Yes.
Or so? Or so. And so, you know, we pick things that we think are of interest, but there's a lot more of interest out there, of course. And so I'm glad that you like that you were picked and that your peers do too. I think that's great. I think it's really a cool thing that we do here. We're very lucky to do it. Okay, now we have a couple of snippets. And I know that we have too many. We have,
yes,
and Alan doesn't like having a lot of snippets, but I'll go through them really quickly. And that's it. What did you think of those snippets?
I like the snippets. I just they're the reason that the show is sometimes nudging past 2 hours, and I know some people are not thrilled with that. And
we will not nudge past 2 hours today. I have to leave at 5 to pick up my son.
We'll just blow past 2 hours.
The first one I heard about when I visited Wisconsin a couple of weeks ago, because the group I visited did this work, and I said, well, I'd like to discuss it when it comes out. And it just was published in MSphere this week. Seroprevalence of Zika virus in wild African green monkeys and baboons. And the first two authors are Connor Buechler and Adam Bailey, who I had beers with one of those nights I was there. And Adam Bailey had previously invited me to Wisconsin. And then David O'Connor is the last author, and Tom Friedrich. And of course, those two were both on the last TWiV from Wisconsin as well. And during that TWiV, I said to them, have you or anyone looked in monkeys in Africa for evidence of Zika virus infection? And they said, well, we just submitted a paper. And that night, while having beers with Connor and Adam, they said it had been accepted. So the TWiV bump in real time, right?
Right. Now, since this is in MSphere and it's open access, and you're the MSphere, what are you, the podcast editor? No, the
eye editor.
Eye editor. So does this get extra links then, or
it gets
something?
In the table of contents? There will be a link to the TWiV episode. Yeah.
Yeah. Cool.
So, you know, it says that means the article was highlighted, and maybe we get a little traffic as well. And we highlight the article, so maybe more people will look at it. And in fact, one could do a study after a year of this and see if TWiV papers got more views than others. Anyway, what they did here is they wanted to look at the prevalence of Zika virus and anti-Zika antibodies in wild African non-human primates. So they got 4 groups, 14 Chacma kinda hybrid baboons. These are 2 different species bred from Kafue National Park in Zambia, 41 yellow baboons from Mikumi National Park in Tanzania, 25 African green monkeys from the Gambia, and 159 African green monkeys from multiple sites across Africa. So they did first unbiased deep sequencing on plasma samples. No Zika virus detected. But they did find lentiviruses, pegiviruses, artery viruses, other viruses. No Zika actively infecting these monkeys, and not even pieces of Zika virus genomes. Then they looked for antibodies. They use an ELISA format, antibody capture, and they had some issues with cross-reactivity with dengue virus antibodies, and so they go through some procedures to try and minimize that. But in the end, what they found when they put their thresholds there and so forth, that 4 of 25 Gambian African green monkeys had evidence of exposure to Zika via antibodies. 2 of 41, that's 4.9%, Tanzanian baboons were positive, and everyone else was negative.
Is it Tanzania or Tanzania?
It's Tanzania.
Tanzania?
Yeah.
And how do you say Tanzanian or Tanzanians? How do you say Tanzanian?
Tanzanians.
Tanzanians.
Tanzanian baboons.
Mm-hmm.
Not Tanzanian baboons?
No, Tanzania.
Are you sure?
Yeah.
Yes.
Because we'll get email.
I've heard people from Tanzania actually say that.
What?
The correct way.
That they're Tanzanian.
Tanzanian
They are Tanzanian.
baboons.
That's right.
Baboons from Tanzania.
Correct.
Just doesn't sit right.
Well,
But I know it's right. I
It
know.
is right.
But I'm just, So not a lot of these monkeys had antibodies to Zika virus.
Right.
Which is interesting because we think that is the reservoir in Africa, right? Remember the first isolate of Zika came in 1947 from a monkey.
Captive macaque.
Yeah, a captive macaque. And the idea being that monkeys are infected and the mosquitoes are spreading it amongst them as well as to people. But
So that suggests that isolation was pretty darn lucky.
Yep.
well, that monkey
I mean,
sat out
unless
there.
there's been...
Yeah, that's right. It probably was lucky. Yeah.
The other possibility is that probably these monkey populations have changed a lot over time. I know there's been a lot of poaching and human encroachment on habitat and all that. So it may be that the chain of transmission is not as robust as it used to be. I don't know.
So say they saw more Zika-exposed animals from northern areas of sub-Saharan Africa, even given the more extensive sampling performed in the south. So there may be geographic limitations imposed by geography or vector or forest conditions, etc., which I think would be interesting to look at, right? People should continue to do this because it would be interesting to know what's there at the moment.
Do you know of any research going on in non-human primates with Zika virus in laboratory?
Excuse me, what did you just ask me?
She knows.
The TWiV I just did in Wisconsin, dude. This group,
And do they find the same effects in the non-human primates as they do in humans?
What kind of effects?
Microcephaly,
No.
shrunken testes.
No. So what they do is they infect them subcutaneously, multiple sites to imitate a mosquito bite.
Right.
They get a viremia.
Yeah.
And no pathology associated with it. They've done it in some pregnant monkeys, and they have seen some pathology in the CNS of the fetuses, but not outright microcephaly.
Okay.
They need to do more of that, of course. But this group has been doing that in Wisconsin.
Okay.
And that's when I asked them, do you know anything about monkeys?
All right.
Tanzania.
Well, I'm going to weigh in on this in a minute. But
weigh in. Go ahead.
Well, I looked it up, and there's a forum on Photos.com, whether it's Tanzania or Tanzania or Tanzania or Tanzania. And it may be that North Americans use Tanzania and the rest of the world uses Tanzania, but it doesn't necessarily sort it out with respect to the accent or how you pronounce the A vowel. Mm-hmm. But one person said most of the Tanzanians I've encountered use Tanzania, and I've only heard the other pronunciation a couple of times. So it was originally from Tanganyika and Zanzibar. Since Zanzibar was pronounced with the Zan, it makes sense to pronounce it with the Za sound. More than you possibly wanted to know.
I wonder if anyone is listening from that country, let us know.
Let us know how you pronounce it.
Wow. Do you know where it is, Dickson, in the country?
I do know where it is.
Where in the continent, I should say.
I was almost in Tanzania. It's contiguous with Kenya.
And Brianne, how do you know how to pronounce it?
I recently started taking students to South Africa, and my colleague who does that is now starting a new program in Tanzania. So I've been hearing a lot about
Tanzania.
his program recently.
All right.
So I get all my pronunciations from attending the American Society for Tropical Medicine and Hygiene meeting, and that's the only way they pronounce it. And so,
And Vincent, if you look at Figure 1, where they have the maps of Africa, the one under yellow, wild Tanzania, that shows you where Tanzania is.
Yeah, I was looking at that. I wanted to know if Dickson knew.
I did.
In
fact, I was there during the time when there was a big dispute going on between those two countries, and there was a meeting held at ILRAD, which was in Nairobi, just outside of Nairobi, and a lot of people from Tanzania were at the meeting, and they couldn't go home.
Okay.
They were stuck.
Well, maybe.
Presume eventually they did get home.
Hopefully we won't have any more papers from that country.
No.
That's horrible.
No, I hope
Maybe you'll have more papers once you figured out how to pronounce it. Yeah.
Yes, that's the
Tanzania? Is it Tanzania?
Tanzania.
I would say Tanzania.
Because I've heard both.
We've covered all bases at this point.
Tanzania. I just want to remember.
We're going to get letters.
No, I need reinforcement. I need reinforcement to learn things. Orzo is not rice. Tanzania is.
There's a Tanzanian out there listening.
All right, so we have two other snippets that have to do with a Zika virus vaccine, which is quite interesting. It's an RNA vaccine. So instead of an inactivated virus or an infectious virus or DNA virus or a virus-like particle, this is RNA, which you may think, what? It's so unstable, but there are ways to protect it. So we have two papers. One is a letter in Nature, Zika virus protection by a single low-dose nucleoside-modified mRNA vaccination. The first authors are Norbert Pardee and Michael Hogan. They both have asterisks, which probably means that they're co-first authors. Yes, they've equally contributed. The last author is Drew Weissman, and these are from the NIH, a company called BioQual, Duke University, which we have a representative of here today.
In fact, members of my undergraduate lab and my graduate school lab are authors on this paper.
Cool.
Wow.
Wonderful. University of Pennsylvania, a company in Vancouver, Kansas State University in the Little Apple. Yeah, there you go. Center for Virology in Germany. Center for Virology at the Deaconess Harvard in Boston. Then we have a company in Mainz, Germany, and then NIH again. Okay, so what they do here is they make a modified, they make an RNA encoding the PRM and E glycoproteins, and they make this RNA with modified nucleosides, which prevents the RNA from being sensed by the innate immune system. Yes. Is that correct?
That is correct.
Does that work?
It can work, yeah.
Okay, it just dampens it, I guess, not 100%. Though,
no,
right?
not 100% at all.
And they also say it increases translation in vivo. Okay, so then they formulate this with lipid nanoparticles, and so that, I presume, encapsulates it and protects the RNA so it's not degraded.
Yes.
Right. And then when you inject those, I assume they're taken up by cells, and then that gives you the immune response. You make the proteins that are encoded in the mRNA, of course, and that gives you the immune response. In the other paper, they actually show that these RNAs give rise to particles, empty particles. They don't do that here. So they do a couple of experiments. They evaluate the immunogenicity in mice. They use C57 blacks, intradermally immunized. They get T-cell and antibody responses. They get neutralizing antibody responses. They also use BALB-C mice. They do the same thing, and they also get nice responses. And in the BALB-C, they do a challenge study, and all the Zika mRNA immunized mice are protected from detectable viremia. These are wild-type mice, so they're not going to develop disease because you have to take away the the interferon system to do that. So a single immunization gives protection. And then they go into rhesus macaques, immunized intradermally with several doses. They all get and these induce glycoprotein-specific antibodies, all 3 doses. They challenge these monkeys, subcutaneously, and here the result is a viremia. Vaccinated macaques are protected. 4 of 5 animals had no detectable viremia, and the other one had a transient blip. A viral blip, they write. That's interesting. That's new, a viral blip. So this looks good. It's a single dose with this, and it's protective in mice and in non-human primates, and I presume eventually this could go into people.
And this paper is also open access. So if you want to delve more deeply into it, you can do so.
Into viral blips.
Yes.
Right. They made the point that both in mice and in macaques, they were getting, at least for mice, it was better neutralizing antibody titers than other vaccines. And in the macaques, also better than a DNA vaccine. So
yeah,
seems pretty powerful.
Yeah, it's not too hard to make. You don't have to grow virus and inactivate it or attenuate it and so forth. So
right. That was a point that I noted when they were talking about the advantages of this just right in the beginning of the paper, that they forgot to say how it's a safety thing because they don't have to grow up the virus.
Right.
So, yeah.
And then there's a second paper in Cell, Modified mRNA Vaccines Protect Against Zika Virus Infection. The senior author here is Michael Diamond from WashU in St. Louis, who we had on TWiV not too long ago. First authors, Justin Richner and Sunny Himansu. And here, again, an mRNA encoding. Now, here the encoded proteins are slightly different. They encapsulate a modified mRNA. Do they modify it with the same...
You didn't put the paper in the show notes, so I haven't read this one.
Okay, don't read it. I'm just going to summarize it briefly. Basically, they make it's a proof of principle also. They express enough viral proteins to make virus-like particles. They show that this is protective in the immunocompromised mice. They also do an interesting thing, which is we know that antibodies to Zika have the potential to enhance dengue infection by antibody-dependent enhancement. So they actually remove the amino acid residues of the glycoprotein that are responsible for that, and they show that in a mouse model you get less antibody-dependent enhancement. In a mouse model for dengue virus, you get less antibody-dependent enhancement, although you also get less neutralizing antibodies against Zika virus as well. So again, it's another mRNA vaccine. They're lipid particle encapsulated, same thing as the other paper. What I wanted to point out here, they have a lovely discussion in the discussion. They say our mRNA vaccine adds to a burgeoning pipeline. 3 groups report DNA plasmid vaccines encoding ME or PRME genes. A rhesus adenovirus vectored vaccine encoding the Zika ME genes. An alum adjuvanted chemically inactivated Zika vaccine that induced neutralizing antibodies. A human adenovirus vectored vaccine that produces soluble E glycoprotein. So all of these have shown promise in animals, but as they say, none of them have been evaluated for cross-reactive antibodies. And they say, we don't actually know if Zika antibodies are going to be a problem for dengue. We don't have any epidemiological evidence, and so without that, it seems hard to justify evaluating this in vaccines for people unless we know it's a problem. But they say that our mRNA platform is cool because we could change it quickly if we needed to. So that's a cool summary of what's going on. A lot is going on with
and that
Zika vaccines.
That paper is not open access.
Yeah, I felt I should mention it because same idea, and it just came out at the same time. Okay, Zika mRNA vaccines. Okay, Dickson, does that work? Does that work for you?
Works for me.
I think that's interesting. I would like to tell you about the sponsor of this episode, Blue Apron. Their mission is to make incredible home cooking accessible to everyone while supporting a more sustainable food system, setting high standards for ingredients, and building a community of home chefs who know the difference between orzo and rice.
Right.
Now, that's not their goal, but...
They've got some work to do.
They do.
What they do is they deliver seasonal recipes along with fresh, high-quality ingredients so you can make home-cooked meals. Every meal, you get a box. You can get it for 2 people or 4. And it comes with a recipe card, lovely recipe card with easy-to-follow instructions, even with pictures. And pre-portioned ingredients, 40 minutes or less for each meal. And if you spend a lot eating out or buying groceries, less than $10 a person. Why can they do this? Well, they don't have stores. Stores cost a lot of money to maintain. The old brick and mortar. And so they have 150 local farms, fisheries, and ranches all over the U.S. that they use to supply their stuff. For example, seafood they get under sustainability requirements developed with the Monterey Bay Aquarium Seafood Watch. They have beef, chicken, and pork from responsibly raised animals, produce from farms that practice regenerative farming. And because they only give you what you need, there's no waste, and you don't overeat. You can customize your recipes every week depending on when you want them. You don't have to get them every week. Whenever you want, you can get what kind of food you want, meat or non-meat options. And they deliver to 99% of the U.S. They do not repeat their recipes in a year. So you get something once, that's it. And so this month's recipes include the fateful salmon piccata with orzo and broccoli. And we should point out that orzo is an Italian pasta that looks like rice. I can't believe we did that. Pork chops and miso butter with bok choy and marinated apple. I'm not going to say anything about any more ingredients. Vegetable chili and baked sweet potatoes with crispy tortilla chips. I've made some of these. They're really good.
So, Vincent, if I like something, can't I order it again? Or it's gone and I have to accept the next?
It's gone.
I think
it's gone.
Don't they do within a
however,
month?
No.
Can you do within a month?
No.
You know about this?
I do. I actually have started using it recently as well. They give you a recipe card with enough information that you could make it again yourself.
You can make it yourself.
That's very nice.
I did this. I served one of their meals was beef tortas, which are basically a Spanish cheesesteak sandwich, and we loved them and said, "Hey, we ought to have those again." So I just transcribed the recipe and got the ingredients locally, because they do ship the ingredients in the exact quantities you need, but they're usually you can obtain most of them, or at least decent substitutes locally.
Right.
Can you reorder within a month though? The same thing, or that's
No.
once only?
No, it's gone.
That's it.
That's it.
It's gone.
Poof.
It's a weekly thing.
Yeah.
Yeah.
But-
Yeah, 6 different recipes every week.
However, you end up with a cookbook when you finish, because at the end of a year, you've got 165 different
I have
recipes.
probably 3 inches high worth of recipe cards from the 2
Keepable,
years
huh?
I've been doing it. Yeah.
Yeah, I am a terrible cook, and I am learning a lot about cooking, and also getting a lot of new ideas.
Oh, good.
Are you a good pipetter?
I am a good pipetter, and I am a good baker, but
A good baker.
Cooking-
No, bakers count. Bakers count. That's difficult to do.
cooking's no good.
Cooking. Oh, gee whiz.
Yeah, so that's what I heard on another podcast that was doing this ad, that you can keep the recipe and repeat it if you'd like. And they said also it teaches you how to cook, as
Right.
you
Yes.
said.
It does.
Right.
'Cause you can, and this is a very easy way to learn and put things together and so forth. Plus, it really is tasty. The food is excellent. I think this is a great thing, and I'm sure there are other imitators, and it's very popular because if you're busy and you don't want to buy things, this is the big thing. I don't want to buy everything, collect it, and then start, and I forgot that.
Vincent, imitators. I think that sounds like a side dish.
It's orzo.
No, imitators.
Right.
I know.
Fried imitators.
Okay, so-
Alan doesn't like your puns.
No, I like his puns,
You do?
but the appropriate response to them is groaning.
It is?
Yes,
that's a
what
high
Kathy
compliment.
does.
That's a very high compliment.
Kathy's Grown. The famous Kathy Grown.
Right.
Check out this week's menu. You get 3 meals for free with your first purchase with free shipping. Go to blueapron.com/TWiV. Blueapron.com/TWiV. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron. So don't wait. Blue Apron, a better way to cook. Did you get into it because of TWiV?
I did.
Oh, it's great. This is great.
I'd heard about it a few times before, but I figured that was the...
Did you use
Thank you. Get me to start.
Did you get your 3 free meals?
I did.
Oh, excellent.
Nice.
What better advertising than to have... You get an A. You get an A.
Oh, thanks.
All right. Now we have another interesting paper, which just came out. The one, there are actually 2. I'll just talk about one in some detail and the other a little briefly. It was published in Current Biology. Structure-function studies link class 2 viral fusogens with the ancestral gamete fusion protein HAP2. Authors are Jennifer Pinello, Alex Lai, Jean Millette, Donna Cassidy Hanley, Jack Fried, and Theodore Clark. And they are from Cornell University in Ithaca, New York, where I'll be in a couple of weeks, actually, doing a TWiV up there. So HAP2 is a protein originally identified in Arabidopsis. Now, here's the story here.
Which is a plant.
Which is a plant, a flowering plant.
Flowering,
right?
That's angiosperm.
So, sex, the production of the reproduction by sexual means begins with fusion of gametes, two different gametes. They're haploid. They make a diploid cell, and then they proceed. And that step, which is so key, is really not well understood. And it's assumed over the years that it involves some kind of fusion protein to trigger the two fusions, but we don't know how it works. So this story begins with a screen a number of years ago in Arabidopsis, which is genetically amenable, to find basically genes which, when you knock out, result in sterility. And they're haploid specific, so that's why the HAP2 gene, I think they identified a number of different genes that contribute to this phenotype. But even though that was done a long time ago, nobody really understood how they work. It's just incredible that, and they've done a lot of very interesting work. This gene is present throughout a lot of organisms, not just plants, but unicellular algae and many others as well. It's what they call deeply rooted in the tree of life. It's been here for a long time, and it's probably was probably around at that common ancestor to all the eukaryotes before all of these organisms branched off. It's that old.
And one of the things about this paper that I kept thinking as I was reading it was, wait, nobody had done that yet?
Yeah, I know.
Wait, we didn't know that? Wait, how do we not know all this? This is really fundamental.
I know. It's funny that it was not done, because once you see what was done, you say, that could have been done a while ago, right?
Yeah, it could have been, but it was not obvious to look here and at this particular thing. And I think it's really cool that there's stuff out there that is really just fundamental to life as we understand it. This is where one of the basic things, as Vincent said, that you need for sexual reproduction, and that's plants, that's animals, that's all these things, and we just don't understand how that fusion step happens. So very cool.
This paper uses Tetrahymena thermophila. Tetrahymena is a well-known species among biologists. Do you have a Tetrahymena person at Drew?
We don't, but we do have someone who does some work with Chlamydomonas.
Okay.
It's the other organism in the other paper.
And Chlamydomonas is an algae.
Yes.
And Tetrahymena is a ciliate, and they're both ancient organisms. They've been around. And Tetrahymena, of course, is well-known because self-splicing RNAs were discovered in it, and that got a Nobel Prize for Tom Cech, but lots of other discoveries as well. And so this protein HAP2 is present in Tetrahymena. In fact, when two cells are joining in the mating, the protein's present at the junction between the two cells, called the conjugation junction. And lots of pores form there for the exchange of material, of course.
Not to be confused with conjunction junction.
Exactly. The function of conjunction junction.
Yes.
And these cells kind of look like orzo.
Yes.
A little bit.
That's right.
Orzo,
They do.
right?
They do. They stick together a lot.
So they have a nice assay where they can take there are different mating types. How many different ones are there? 11 or 7? Seven different mating types. And they can't mate with each other, but you need a different mating type, and you mix them together and they will fuse.
Right.
And then they load these cells up with two different colored dyes.
Boy, their bathroom laws must be really screwed up.
They have a single entrance
Oh my
and
God.
15 exits.
Truly, yeah. They load these gametes up with amine reactive dyes, a red one and a green one, so they react with proteins, and then when they fuse, they can trace the movement of the protein.
So clever.
Isn't that nice?
Yes.
Very clever.
So they can see the contents exchanging readily. You can see these under the microscope.
It's beautiful, too.
That is
The
very
illustrations are lovely, which this is not open access, by the way. I know I'm kind of keeping track of this. But
So basically, if you take away the HAP2 gene, you can easily delete it. You don't get exchange of labeled protein. They don't exchange contents when you mate two gametes. And so they use this system to study the kinetics and so forth. And it seems to be that if one of the pair is hap2 deleted, you still get pores formed, but they're not as efficient. So these cells, you have to have hap2 on both of them, hap2 protein on both cells, both gametes, in order to get very efficient mating. So they develop a cool flow cytometric assay for looking at the exchange of material between the two cells. And basically they confirm that you get good protein transfer in the wild-type strains. If you have hap2 deletion in wild-type, you'll get transfer, but it's not as good as with the wild-type crosses. And you can complement the defect by adding back the hap2 gene. So hap2... Go ahead.
I want to just step in because what you said about having it expressed on both cells, the rates at which they fuse is reduced when it's expressed on only one cell, but they can still form fusion pores.
Yes, they can still make forms, but there's less, right? Initiation of pore formation is the primary defect in the heterozygous crosses, if you will. But so they still can exchange a good amount of protein, even though they have fewer pores, right? Because they say the overall amount of protein exchange in pairs that were capable of fusion is unaffected. So what is going on here? They take hap2 now and they use computer modeling programs to say, what does it look like? And this is one of the things that a person here named Barry, what's his name, Dickson? Barry, the protein structure guy here. Barry Honig? Is that his name?
Barry Honig, yes.
He's developed, I think one of these might be his program, where you can put in a protein sequence, and it tells you what this looks like, and it compares it to other proteins. So it turns out this thing looks very much like a class 2 viral fusogen.
These programs, by the way, are really cool.
Have you run them?
No, I have not run them, but I did an article on structure determination, and this is kind of a real long-standing challenge in structural biology, because you look at DNA sequences and you can figure out protein sequences that are encoded by the RNA. But then you say, well, okay, this is all the information the cell needs to fold this protein into a three-dimensional shape. Can we do the same? Why can't we just predict the structure? And it's been really, really hard to do. And so these programs they used here, Fire2 and RaptorX, they use things like machine learning algorithms and huge databases of structures that have been solved. Wow. And they say, okay, computer, this sequence makes this protein, and this sequence makes that protein structure, and then you give it a sequence whose structure hasn't been solved, and it tries to predict what you get. And if you plug this into a couple of different models, you often get slightly different answers. But when they converge on something, that's a pretty strong indication that you're going to get that type of a fold when you put the protein together.
Right.
So they pull out with this hap2, the type 2 fusion proteins, which are on dengue virus, Zika virus, and many other viruses. There are type 1, type 2, and type 3 fusion proteins. Type 1, the classic, is the influenza virus hemagglutinin, which is mostly alpha helical. It's perpendicular to the membrane. It is a trimer. The type 2 are parallel to the membrane. They lie flat on the membrane. They're mostly beta strands. And they're typically dimers on the membrane of the virus, but when they fuse as trimers. So this looks like this type 2 protein, and it has a fusion loop, which is where the business end is. The fusion loop in the virus, of course, is inserted into the endosome membrane upon acidification and leads to fusion. So this protein looks very similar. So now you can do experiments and say what parts of this protein are important. And they do some of that. So they delete the fusion loop. What do you think happens to gamete fusion when they do that?
You're going to ask me that, aren't you?
Dickson, what do you think happens?
I don't know, Brianne. What do you think?
I think fusion might not happen.
Yeah,
it might get confusion.
Which is beautiful. It confirms. And in fact, that protein is produced in cells, right? Because you could argue, the deletion doesn't lead to protein production. They check it. Now, deletions elsewhere in the protein make it not expressed well, produced well, but this one is produced well and it doesn't fuse. So the loop seems to be important. They alter specific residues as well. and it changes it. And in fact, there are two cysteines that bracket this fusion loop that are important, and change those, forget it. So the fusion loop is important. Now, what's interesting is the fusion loop of, what did they put in, dengue virus? Yes, they replaced the HAP2 fusion loop with dengue. It didn't work. That would have been so cool.
That would have been fantastic.
That would have been really cool. But maybe they should try some other ones. But then they try and make a pseudotyped virus. They try and make VSV with the HAP2 protein.
To see if it gets...
And it's not infectious.
Darn!
Isn't that too bad also?
That would have been a lot of fun too.
Those are two great experiments that I really like. And then they do some biophysical experiments to show that what this loop is doing. So first of all, they use circular dichroism spectroscopy to show when you mix the fusion loop peptide with membranous vesicles, it becomes beta strand. It becomes mostly beta strand, which is what you would expect, which was what happens with dengue. They also use electron spin resonance to show that if you add the peptide, the fusion loop peptide, to lipid mixtures, it becomes oriented in a way that's consistent with peptides inserting into a lipid bilayer, if you will. So it's doing all the right things. And finally, they do some lipid mixing assays. So they have a lipophilic dye, and they have lipid vesicles. And if the fusion occurs, the dye becomes dequenched and fluoresces. So fluorescence is an indication of fusion. And they show that this peptide causes fusion among vesicles, just like the dengue and the influenza virus peptides do as well. So it seems to be that this protein that mediates gamete fusion in lots of different organisms, all the way down to modern day, is related to a type 2 viral fusion protein. Now, the other paper, before we sort of discuss the implications, I just want to tell you, in the other paper, also a Cell article, the ancient gamete fusogen HAP2 is a eukaryotic class 2 fusion protein. Here they study Chlamydomonas HAP2. They do a lot of similar experiments. They show that the loop, they first model the protein, they show the loop is homologous to the fusion loop of type 2 proteins. They alter it and show that the altered proteins do not lead to fusion of gametes in Chlamydomonas. But then they produce the protein. They show that it inserts into lipid vesicles in what looks like a trimer. And then they solve the crystal structure of the trimer, and it looks just like the trimer of the post-fusion trimer of the type 2 viral protein, which is, again, after acid catalysis, after acidification of endosomes, these type 2 viral fusion proteins trimerize and reach up, but the fusion protein gets stuck into the endosome membrane. And this HAP2 protein in membranes is already a trimer. And you can see it looks just, the topology is very much like that of the type 2 protein from viruses. So it's really interesting that, so this is a post-fusion configuration which didn't require acidification, which makes sense because as far as we know, fertilization doesn't require acidification, right? And so this is happening at neutral pH. Somehow the membrane is triggering the trimerization and the exposure of the fusion peptide. So that's going to be very interesting to sort out. And this is just beautiful. I like this structure paper a lot. It's got, they're lucky they have the structure. The other paper had nice experiments, but this one has the structure. And so they say these proteins are so similar, there's no way this is convergent evolution. This is an ancestor that got handed down. And, even if this HAP2 or a related protein was present in the last ancestor of eukaryotes, viruses were around before then. So probably viruses gave it to eukaryotes, who then passed it down here. So that's an interesting idea, right? That all the sexual reproduction that's been enabled by these proteins came from viruses.
Some early syncytial virus was used this as its means of transmission, and that got incorporated into a cell gene that could then be used to fuse sex cells.
They say here in this first paper, a key step, if not the key step in fertilization, was made possible by a virus. Without that, sex and the diversity of life that it spawned, including man, may never have evolved. It's just great.
Well, and interesting. Go ahead.
I was just going to say, a little bit in contrast to that is when, Vincent, you were talking about all the lineages in which this HAP2 gene is found, deeply rooted and so on. It's not found in fungi and vertebrates.
That's right.
So what they say about that is such sexual species have clearly found an alternative means to accomplish gamete membrane fusion. But whether they use novel proteins or variants of currently known fusion proteins remains to be determined.
Right.
So in fungi, we don't know what kinds of proteins are doing fusion, or they're just not HAP2-like. Is that what they're saying?
I think what they're saying is that fungi lack HAP2 orthologs.
But there's something else that must be doing it, right?
Right.
And so that's probably got some homology to fusion proteins, or maybe not.
So when protozoans conjugate, is there an actual fusion of their membranes?
I don't know.
Or does the DNA simply stick itself through some pore that's created?
They
for
fuse?
The moment?
The protozoans fuse.
Yeah, that's called conjugation.
And the other mating types?
Yep.
So it's basically sexual reproduction, right?
Well, they wouldn't call it that, though, because they both exchange material. So,
They're not haploid, both sides?
no, they're both diploid.
Oh, I see. I don't know. Do you know, Brianna?
I don't know.
Because, I think there are a lot of people out there that might take exception to the fact that says that viruses facilitated the development of sex. But you don't know the age of that protein, so you can't really say which one was there first.
Yeah, but we know that viruses predated the last common eukaryotic ancestor.
Understood, though,
right?
but protozoans were there way before that, and they were conjugating like crazy. See, so that's what I'm saying.
They were there before the last common eukaryotic ancestor?
Not the eukaryote ancestor. They were there before sex in the eukaryotes.
I understand, but they're still a eukaryote,
Oh,
right?
of course.
So they
Of course.
they post-date the Luca, or Lika.
Yeah.
You will.
Or chlamydomonas or chlamydomonas, either way.
Yeah. No, this is a theory, of course.
Of course.
It's just, it looks very compelling if you look at the data, because viruses are ancient, and these... now, the one thing that's interesting that we should mention, and let's see, what paper is it in? Okay, it's in the Cornell paper. They mentioned basically that the viral fusion proteins, although they evolved from a common ancestor, coding elements for these proteins arose independently in different viral lineages through the capture of a cellular gene.
Through the capture.
So their idea is that it started in a virus and went into ancient eukaryotes and then back into viruses.
I see.
It seems a little bit complicated to me.
Yeah, it really does.
Isn't nature parsimonious usually?
Usually.
Right?
I think so, yeah.
You would think so.
But they also, it's highly experimental too, right?
Yeah.
Yeah, of course.
So you have a lot of colonial...
What was beneficial at the moment.
That's right. You have a lot of colonial protozoans as well, like Volvox, for instance. So they have to fuse in order to become polyclonal, basically.
Right.
So maybe that protein was used for that purpose. Rather than have sex, it's just
Yeah,
get a whole
sure,
bunch of cells together. Yeah.
Of course, this is not the only example of the invasion of eukaryotic genomes by viruses. We have syncytians that are needed for placental development coming from retroviruses.
RAG 1 and RAG 2.
Really?
In the immune system, yeah.
Where'd they come from?
They came from a transposon that someone someone actually found the transposon and looked at the virus this summer.
Wow. Well, we need to do that paper.
It was really cool.
That's a cool.
That is
My students
cool.
really liked it.
Oh, I didn't see that. Then there's, we did a paper on TWiV from Nels Eldy's group where the enhancers for interferon-stimulated genes are retroviral. Like 14% of the enhancers come from
That's
endogenous retroviruses. It's just amazing.
It is
Right?
incredible.
And that's what we've found, but there's more in there, and I'm sure there's more. So that's pretty cool.
So you'd call that lateral rather than vertical evolution, and transfer from this way instead of through the germline. Yeah. Lots
But
of
taking a
biology.
gene from a virus and using it is called exaptation, I believe.
Exaptation, I like it.
Exaptation. Yeah.
Named
That was a
after-
new one when I first read it with Nels.
It's named after Frank Zappa, I presume.
No. So, Dickson, what do you think? Is this neat?
It's fantastic.
As the outsider here.
No, it's fantastic. It's fantastic. Better than neat.
Because you're really the politician, right?
If you do that, half of your listeners will try to get rid of me.
Do you think you buy this, that viruses led to sex?
It's not conclusive, so therefore, no.
What would you need? You want to go back in a time machine?
Well, There are other reasons for cells fusing rather than having sex. That's what I'm saying. So this may have been there prior to the arising of sex.
Mhm.
Right.
Yeah, of course.
So if that's true, then maybe it was good just to allow cells to fuse together to begin with.
So you're this protozoan fusion that you're talking about would be an example of that.
Exactly.
So I bet they know what proteins are important for that fusion, right, in protozoans.
You think?
I don't know the
Well,
literature, of course. I don't read that, but
Right.
that's why working in a biology department, if you have protozoan,
Yeah, that's right.
It's
people.
working in a biology department and getting to hear about a diversity of things. It's fantastic. I know a lot more about invasive species and neurodegeneration than I ever did before.
yeah. And that's here on TWiV, we learn about viruses that we wouldn't normally read.
Part of the
right?
joy of my early training was to be at Rockefeller University and as a fly on the wall for the first 6 months. Well, I didn't do any single experiment. I just went to a seminar after seminar after seminar. And the diversity of those was just astounding. Just astounding.
I think this is a cool example of how you can take these model systems, right, both Chlamydomonas and Tetrahymena
Yep.
model systems, and you can use modern genetic methods to address these questions that you first say, oh, look at this protein, it looks like this, and now we can modify it.
Right.
I think it's just amazing. It's just great.
I mean, the acquisition of cellular organelles is thought to have arisen through a fusion of sorts between membranes, right? I mean, you get nucleus and mitochondria, et cetera.
The nucleus is a viral invention, maybe.
Is it now?
It's a nucleus-like structure has been found in some bacteriophage-infected cells now.
Really?
Yeah.
Oh, no, I do remember
And
you telling
prions have now been found in bacteria.
My goodness.
What did eukaryotes invent?
Nothing.
So I'm trying to think.
Politics.
I think
We just stole it all.
And Orzo. They invented Rozo.
Orzo.
would it be fair to say
But then they didn't know what
that,
they invented. They were just stuck with this. What is
so
this stuff?
eukaryotes didn't invent sex because bacteria do sex, right?
Yeah, that's another.
Didn't they invent protein-based immunity? Protein-based immunity.
Protein-based immunity.
You know, like antibodies and T cells.
Sure.
You mean
As far as we know.
As far as we know, I cannot think of. I guess I can't think of any protein-based immunity in prokaryotic cells.
Well,
restriction enzymes.
Yeah, but that's the thing is I was deciding whether Cas was a protein.
It is.
Right.
It's a hybrid. It's a hybrid.
Right. Restriction enzymes are proteins, but you're meaning secreted
Acquired.
proteins that
Right.
bind to pathogens
Adaptive.
outside.
That's true.
Yeah, because, you know, interfering RNAs and CRISPRs, proteins are obviously involved in that. But,
Right.
And there's forms of immunity, but they're mostly nucleic acid based.
Exactly.
That's the effectors, right?
Yes.
The end result. But protein based is secreting antibodies, right? And,
That's right.
and so forth. I mean, maybe we haven't found it yet, but that's the one thing I could think of that eukaryotes invented.
Adaptive immunity.
Of course, they also invented. No, they didn't invent cheating either.
Cheating.
Cheating has been around forever.
Cheating is bacterial.
Yeah, bacteria
is.
cheat all the time. And so we did a paper on TWiV last week about how populations of bacteria deal with cheaters. They kill them.
Yep.
Mm-hmm.
Yep.
They bloody kill them.
My goodness.
And we're much kinder as
pretty
humans.
harsh.
Yeah.
Sometimes we elect them to.
So Michelle Swanson, Kathy, did the paper where they basically use a type 6 secretion system. And she said, it's like a spear. You know, they're spearing each other and killing each other. And I said,
Wow.
we don't. Do we? Would you say police are similar equivalents? And she said, well, they don't use spears, do they? I know.
Right.
That's not what I meant.
Well, Pseudomonas uses cyanide.
Is that right?
Yeah. Yeah. There's a system in Pseudomonas that's part of the. It's kind of like quorum sensing system, and if cells cheat on it and are reaping the benefits of
They kill them.
adjacent, systems, then they're more likely to not be producing the antitoxin against
Sure.
this, and they end up cyanide poisoned.
Amazing.
And I think all the antibiotics that are secreted by soil microbes to make a zone for themselves, those are.
Yep. All right, let's do a few email here. This next one is actually a follow-up to last week, but I didn't want the follow-up to get too long at the beginning there. It would be top-heavy and full over. It's from Jory, who writes, Hey, TWiV Voyagers. This is in response to Rich's comment that the differing divergence patterns of CIF-A or CIF-B compared to core Wolbachia genes implies that the selective pressure driving the evolution of the system were likely acting on the prophage. I'll remind listeners from last week. Cytoplasmic incompatibility factor, CIF. These are two Wolbachia phage genes that control the ability of Wolbachia to cause cytoplasmic incompatibility. I took a look at the paper, and while the analysis did show this, it also showed that the divergence pattern for a phage structural protein was different from those of CIF-AB. Thus, it seems the differing divergent patterns are gene and not organism specific, though I'd expect correlation to some extent within a genome. So I don't think you can say which organism the selective pressure is acting on based on such a difference. Please let me know if I'm missing a key point in yours or the author's argument. Right. So I emailed this to Seth Bordenstein, the lead author, the PI on that paper. And he wrote back, correct, the divergence is gene-specific. Even within the phage WO genome, from a functional perspective, selection affecting cytoplasmic incompatibility benefits both phage WO and Wolbachia, since the CIF-A and B genes ensure the spread of both phage WO and Wolbachia through the host population. The arthropod host actually suffers a fitness cost, as the reproductive success of uninfected females is reduced relative to infected females. The phage WO Wolbachia can thus be considered selfish elements that spread by imposing a fitness cost on the host. That's why I emailed Seth, because I wouldn't have come up with that. All right, so Jory continues. My 2 cents is that the selective pressures driving the evolution of this particular system act on the holobiont. The function of this system appears to be to ensure the propagation of Wolbachia, so it seems there must necessarily be a selective pressure acting on the host, which of course is a selective pressure acting on the prophage as well. It needs a place to live. I'm not sure you can really subdivide which component of the holobiont feels the selective pressure in this case. Seth, through the simple lens of cytoplasmic incompatibility, selection favors the spread of phage WO and Wolbachia. The host, on the other hand, experiences a fitness cost because the incompatibility generated between infected males and uninfected females. This is a classic case of genetic conflict between the microbial entities and the host. Jory continues, on a somewhat related note, I keep running into an issue when trying to imagine how this system could have evolved. Namely, that it only works when most of the males are already implementing it, i.e. females only require Wolbachia infection for reproduction when males are providing CIF-A-B in the sperm. In that case, it is a self-sustaining system in that CIF-A-B expressing Wolbachia drives selection of their own retention by killing progeny of uninfected mothers. However, if only a small number of CIF-A-B variants were present, it would be highly unlikely that one of them would infect a male arthropod that would go on to breed with a female arthropod variant that preferred to keep the Wolbachia, and extremely unlikely that it would be enough to kill all of the progeny of females that lack this tendency. Thus, it seems you would require the result of the selection to perform the selection. Does anyone have any idea for a possible scenario that could explain the initial propagation of this system? Is it likely that the system arose out of genetic drift and could then sustain itself? Mm-hmm. Seth, astute point. Wolbachia with prophage WO genes CIF-A and B must drift above a threshold frequency before it can deterministically spread by CI. In its simplest form, the threshold frequency is determined by the overall fitness cost to the host. Thus, if Wolbachia phage WO cause a 3% fitness cost on the host, they must drift above a 3% threshold frequency in the population before they can spread to fixation. Back to Jory. Also, would any of you care to speculate about the Wolbachia WO factor that suppresses the embryonic lethality? That's just something we didn't talk about.
Yeah,
that's an interesting question, right?
Yeah.
I'm wondering whether it could be any old Wolbachia gene, or if it must be one specific to the strains that use the embryonic lethality strategy. The latter case seems unlikely given the author's search for genes specific to such strains returned only CIF-A and B. I wouldn't expect that oocyte-derived CIF-A-B could suppress the effect of those same proteins derived from sperm, but it's technically possible that they could interact differently with the chemical environment of the sperm versus the egg. An experiment where CIF-A-B transgenic males were crossed to CIF-A-B transgenic CIF-A-B females would have been nice. He's acting like a reviewer here.
Yes.
Seth, another great thought. We have preliminary results that there is no self-rescue between the two, but we have more work to do before we're ready to make a firm conclusion. Back to Jory. I think it's more likely that the suppressor is a common Wolbachia protein rather than CIF-AB, but this also raises questions about how this system could have evolved. If there was a subpopulation of rare variants that had acquired CIF-AB, why weren't they simply diluted out through matings with non-CIF-AB Wolbachia-containing arthropods? If the suppressor is a common Wolbachia protein, there would have been no selective pressure to force reproduction, specifically from CIF-AB females, which would be required to propagate these alleles since the female arthropod transmits the parasite. Yeah.
Right.
By contrast, if CIF-AB were indeed the suppressors, initial rare variants would have formed an instant bottleneck, as they would only be able to mate with each other. Could that explain the evolution of this system? This also seems like an easier path to speciation, but it's quite biochemically dubious. Love to hear your thoughts on this. Thanks for the great podcast. Jory in Montreal, Quebec, it's cold here.
Right.
Yeah, these are great points. How this evolved and it got started is really an interesting problem, but because we're very far down the evolutionary line here and only have a little bit of information. So I don't know, but some of those Seth's doing some experiments, so stay tuned. We'll see.
Sounds like they both need to collaborate.
It does.
I mean, they know that literature pretty intimately.
Yeah. It's kind of cold in Montreal.
It's kind of cold in Montreal.
Dickson, can you take...
I would be delighted to. Diane writes, "Hi all. This is my second attempt at a book giveaway, and I hope I'm more successful this time around. TWiV and TWiM are still two of my favorite podcasts. I'm still hoping to be able to add more to my ever-expanding playlist. I'm looking to keep one step ahead of some very bright undergraduate students in my microbiology course here at Lewisburg, and your discussions definitely help me with that. The last time I wrote, we were expecting wintry weather. It has come and gone, and we were anticipating a return to the balmy 60s in the next few days. Keep up the great and lively discussions."
I presume this is Lewisburg College, right? Which is North Carolina, right?
Yes.
You must know that one, right?
I have heard of it, yeah.
Yeah.
And I agree, listening to the podcast does help keep you ahead of some bright undergraduates.
Unless they just pay attention to my comments. And I'm setting the whole world back to the ancient times, as it were.
Now. So which book giveaway is this?
That's an old one.
Right.
Oh, okay.
I don't know. Right now, we're done with book giveaways. We're trying to wait a few weeks before we start a new one and clear it out. Kathy, can you take that next one, please?
Sure. Jen writes, "Hello, TWiV team. Long-time listener, first-time writer. I began listening when writing a paper about autism. I'm an elementary school teacher for Chicago Public Schools. I was looking for active discussion, debunking the myths around vaccines, and you folks fit the bill. I enjoy your discussions of virology. The way your podcasts intertwine the papers, politics, and real-world discussion is both user-friendly, informative, and fun. I frequently find myself citing information from your podcast when talking with parents and coworkers. Keep up the great work. The weather in Chicago is a balmy 29 Fahrenheit with some flurries.
Yeah, that's right.
I hope I win, Jen."
That's balmy for Chicago.
Yeah. The comment where she says she finds herself citing information when talking with parents and coworkers is a frequently heard comment about NPR. But another phenomenon of NPR is driveway moments. And I've never heard anybody talk about a driveway moment with TWiV.
Oh,
well.
That's when you get home and you have to sit in the driveway listening to the rest of the show.
Oh,
Right.
yeah. We've had parking lot moments.
But see, the podcast isn't subject to that because
You can just pause
You can
us.
take your
and
phone
continue
inside.
listening.
you can
Right.
pause and listen. But the radio show, it's gone, right?
Usually it's the moth and they're halfway through a story or something like
Moth.
that.
Yeah.
You like the moth?
I love the moth. Yeah, it's great.
Well,
I like the butterfly also.
I got into podcasts because I got tired of listening to NPR.
Really?
Yeah. I didn't like a lot of their stories, and I was really tired of their fundraising. Of course, look at me. We're doing ads.
Yeah, we're doing ads now.
Could skip through them. Who read that? Kathy?
Kathy read that.
She did.
Take the next couple, Alan.
Sure. Tim writes, "Hey, TWiV friends. You said to write in immediately to get the contest done quickly, so I'm doing so. It's a pleasant -9C early morning listening to you folks and feeding my dairy cattle."
Love it.
Well,
That's why it's in there, because I just
yes.
had to...
Thank you, Tim.
Yeah, right.
We missed it.
Sorry.
Jess writes, "Hello, everyone. Hello again from Philadelphia. I was unsuccessful in winning a previous book, but I'm determined to be the 27th email. I would love to hear as much as I can about new topics in science, since I am currently deciding whether to go for my master's and what to study. Maybe one of your books will give me the inspiration I need to pursue my next degree. The weather today in Philadelphia is pleasant for February. We have mostly cloudy skies with a high of 48 Fahrenheit and a low of 32 Fahrenheit, though it feels much colder with the wind blowing in between all the buildings. Thanks for all you do, Jess. And I'll go ahead and take the next one, too, because those are both short. Bib writes, I happened across this article today, and provides an article from, what is this? It's a veterinary American Animal Hospital Association. Border Collie collapse, or BCC, is recognized as a form of exercise intolerance in border collies, kelpies, and related breeds in the United States, Canada, and Australia. Dogs with BCC are normal at rest, but after 5 to 15 minutes of strenuous exercise can develop incoordination and altered mentation. Recently, JAHA posted an exercise study on dogs with BCC in a particular issue, and a survey on observations of dogs with BCC in the subsequent issue. It struck me that the exercise intolerance here seems to resemble the description of exercise intolerance associated with ME/CFS. Animal model? Which is very interesting.
Yeah, I didn't know about this at all.
Yeah.
So I emailed Suzanne Vernon, who knows a lot about CFS, and she wrote, looks cholinergic to me. If that's true, then it could be a model, since antibodies to cholinergic receptors have been detected in ME/CFS. Okay.
So they could be barking up the right tree. Thank you.
Oof.
Tubby writes, while I'm sure I'm not writer 27, I can at least bump you one closer to getting emerging infections sent on its way. I appreciate your podcast. Try to make sure I squeeze in TWiV, TWIP, and TWIM. Be nice to Dickson.
There you go.
Why?
See, I'm not the only one that says that.
Let's do a couple more. Dickson.
Marianne writes, good day, TWiV hosts. I love to be the winner of your latest book contest on emerging infections. This semester, I am one of the instructors of an undergraduate virology course, and the topic I selected was the emerging viral infections. Thus, this book may come in handy. As always, I put in a plug for the Twix podcast in my lectures. I would also like to put in a positive review for the writing of Marv Roach, who you also mentioned you had on your shelf. I've read most of her books, and she takes a very fresh and funny look at the science around some pretty interesting topics, such as the use of human cadavers in research, stiff, sex research, bonk, food science, gulp, I love that title, and space exploration, packing for Mars. I have yet to read Grunt, but it's on my list. Have a great day. I presume Grunt is about doing just a hard day's work.
Maybe.
Just like
I think it's about military science.
Yeah. We have a book at home which is about milk cows and how they're raised and everything else, and the title of the book is called Moo. I love that. It's so direct. Moo.
I do have, I have Gulp there on the shelf. I think I have Stiff. Maybe I should give them away.
Maybe.
I don't know. We'll see. In a couple of weeks. Kathy, can you take the next one, please?
Sure. Anthony writes, and it's about the legendary Ursula Le Guin on alternative facts. She's a writer, and this is in the Oregonian, and this is the content of her letter. Compares a recent letter in the Oregonian compares a politician's claim to tell alternative facts to the inventions of science fiction. The comparison won't work. We fiction writers make up stuff. Some of it is clearly impossible, some of it realistic, but none of it is real. All invented, imagined, and we call it fiction because it isn't fact. We may call some of it alternative history or an alternative universe, but make absolutely no pretense that our fictions are, quote, alternative facts. Facts aren't all that easy to come by. Honest scientists and journalists, among others, spend a lot of time trying to make sure of them. The test of a fact is that it simply is so. It has no alternative. The sun rises in the east. To pretend the sun can rise in the west is a fiction. To claim that it does so as fact, or, quote, alternative fact, is a lie.
There you go.
A lie is a non-fact deliberately told as fact. Lies are told in order to reassure oneself or to fool or scare or manipulate others. Santa Claus is a fiction. He's harmless. lies are seldom completely harmless and often very dangerous. In most times, most places, by most people, liars are considered contemptible.
It's wonderful. It's wonderful.
Yeah. So you can see why she's a legendary writer.
Yes.
Yeah, it's true. That's something we talked a little bit about, probably got some people angry about. And in science, we appreciate facts.
If this shoe fits, you better start to wear it.
What does that mean, Dickson?
It means that there are liars out there that are in place right now in our government, and they know it, and they should read this and change.
Yeah, but they won't. They won't. But
it's what it means.
You know, scientists says, I think that's what we discussed a while ago that got some people angry. Scientists don't. We live for facts, and this idea that there are alternative facts is just contemptible, right?
It really is.
By the way, Brianne, I saw. We get a lot of emails from undergraduate educators.
I think that a lot of the podcasts are very useful for us to listen to as undergraduate educators. For me, I sometimes miss kind of having the big group around me of immunologists and virologists that I had in grad school. I miss having a journal club that I could go to with like-minded scientists. And so listening to TWiV kind of gives me that option once a week where I can at least pretend like I'm listening in.
Well, you can always come here on a Friday if you
thanks.
want to. You're welcome.
Skype in. Come on.
Yeah,
you can Skype in too. That's right. All right. Time for some picks of the week. Alan, what do you have?
I have something that I just bought recently, but probably many of our listeners have heard about this thing for several years. This nonprofit foundation that developed this thing has been making them for 5 years now. It's called a Raspberry Pi. Not the kind you eat, but the small single board computer that's it's roughly the size of a pack of playing cards. And the only reason it's that thick is because of the USB ports and the Ethernet port on it. The board itself is actually a good bit thinner. I just got one of these and set it up because my daughter was complaining that she didn't have her own computer. Now, before you think that I'm neglecting my child, this is a child who owns an iPad and an iPhone. So, by the standards of me at her age, she's computationally just way beyond. But she made the rather good point that there are some things, particularly some of her homework assignments, where it's useful to have a larger screen. So I didn't relish the thought of buying a desktop just for her to use Google Docs. And this was really an ideal solution because it's, first of all, it's $40 to buy this complete computer on a board. Doesn't use a hard drive. It uses a micro SD card like you would put in a camera. So you install the operating system, which is Linux, on the little card in any other computer, and then you pop it into this single board computer. You plug it into the wall. It has a whole bunch of advantages beyond just the price. You can do all kinds of interesting hacking with it. It's got input-output pins that you can access to put it into a robot or a drone or whatever other project. People have been doing just amazing things with this for 5 years now. And Dickson will appreciate this. Instead of buying a whole big box and power supply and all these resource-intensive things, you're buying this single little board that draws like 5 watts when it's operating at full tilt.
Nice. Very nice.
And I had a monitor and a keyboard sitting around gathering dust, so just plug those in, and it's all good to go. And I am frankly amazed at its performance. It's really, really nice. It's a functional desktop computer. I wouldn't try to edit live video on it or anything like that, but for the tasks that my daughter or, frankly, 90% of people who are using desktop computers do, it's perfectly adequate and just a really, really cool thing.
So you hooked up a monitor to this, right?
Hooked up a monitor and an old Apple USB keyboard.
And she's doing Google Docs on this, right?
She does Google Docs on it. It comes the operating system that you can install any Linux operating system on it, but they've got one prepackaged called Noobs. Which is an acronym for something, but of course it's for newbies to this particular platform.
Yeah.
And it's a Debian-based Linux flavor that gives you a desktop, and there's a browser it's the open source version of Google Chrome, and a bunch of other things come with it. And of course you can install other things. She needed to print something, and it did not do that by default. So guess what? She got to learn how to use the Linux command line.
Great. How old is she?
She's 11.
Jesus.
So this did you put it in a box to shield all the electricity things?
Yes. So for hers, I put it. You can get various little cases. The one that I've linked to is a site called Adafruit, which they're based in New York City, and they sell these, but you can buy them from different vendors. CanaKit is another one in the U.S., and there are several vendors overseas. And also on the Adafruit site, you'll find cases and little plug-in wall warts to power this thing, and all kinds of accessories depending on what you want to do with it.
Amazing. Yeah, people have been ranting about this on the tech podcast.
Yeah,
this is another generation. It's got a gig of RAM. It's got an ARM processor, which is similar to what goes into some of Apple's products, right?
Right.
Amazing.
Yeah, it's a really, really cool little device.
Wow.
Yeah, it's pretty neat. Dickson, what do you have?
Well, I've got a dedicated website to our current EPA director, Scott Pruitt. It's a NASA website that shows various images collected over time to, unless you don't trust the dates that they were taken, which is the only thing you could possibly question about this, it shows indeed that things are warming up and that glaciers are retreating and ice sheets are breaking off and becoming icebergs, et cetera, et cetera. So if you can look at change over time, that's the definition of evolution. If you don't believe what you see, then you obviously should go to an optometrist and have another pair of glasses made for you. I checked up on his background to see whether he'd had any science or not ever. There's no way to know if he did at Georgetown College, which is a small Baptist-oriented school. But he went to law school after that, and I don't think there's much science taught in law school. So he's becoming the head of the agency, which is the gatekeeper for all the protection to our environment, and he refuses to accept the fact that CO2 is a greenhouse gas and that it's making the atmosphere warmer. So I'm very depressed over this. And,
this website has a slider. You can pull the slider across and see before and after.
Really cool.
That's right.
I like it a lot.
So you tell me...
You can flip through different images, satellite images, and you can show... Yeah.
You know, and there's a movement, I've heard this, I'm not sure that this is going to be fact or not, but I've heard the rumor to the effect that they want to discontinue the Landsat program at NASA. They want to defund it. And the only reason I could possibly think of for wanting to do that is that, of course, you'll see things that you don't want people to see. Yeah,
they're defunding every agency that is providing data on climate change.
That's right.
Which is actually, frankly, terrifying in some cases. They are pulling funding out of NOAA, which, as I commented on Twitter, should terrify anybody who flies.
Yes.
Right, so this is where your weather forecasts come from. I don't know what we'll do on TWiV when we don't have those, but
we'll just make them up. We'll make alternative facts. Come on, Alan.
Yeah, so they're pulling funding from all the organizations that have told them things that they would rather that their donors would prefer not be heard. So, yeah,
Dickson, the EPA had under Ronald Reagan. What was his name?
Good question.
I forgot, but anyway,
that wasn't Whitman.
No, but anyway, he wrote an article in the Times this week saying how they tried to do a lot of this back then, and he said the public rose up in arms. He says when you push them to a certain point, when the air is going to be filthy and the water's not good, then they'll yell. So. You know, we may have to get to that point. But
by the way, it's so ironic to look at the country that we used to point to and say they are the people that we're trying not to be like, and that is China. China is now leading the world in alternate energy sources other than fossil fuels, and they have a saying now in Chinese that says someday soon the air will be blue.
And they are actually finally starting to take their very, very serious air pollution problem seriously.
Exactly.
At the governmental level. They've
had to say. Yeah.
Epic smogs, and
that's right.
And it has now reached the point where their government is finally taking at least some of the actions that they need to. So yeah, there's hope there.
If you want to lose a war of wills and a war of economics. Go up against China sometime and don't do what they're doing, and you'll end up on the back seat. That's for sure.
So I misspoke. The EPA administrator in the Reagan administration was Ann Gorsuch, of course.
Ann Gorsuch.
This must have been a subsequent person. But
she's the mother of
Supreme Court nominee.
That's right.
No, you're
Exactly.
kidding.
No.
Yes.
No, he's the Supreme Court nominee.
He went to Columbia, you know.
I know.
Columbia grad.
A lot of people went to Columbia.
Yep. Kathy, what do you have?
Well, as an academic, having been at several universities, I get alumni magazines from those where I am an alumna, and also from those that I've just associated with, such as University of Georgia, and I get sort of the equivalent of an alumni magazine from the U of M. This came from UC San Diego, an article about a cognitive scientist. So that's the connection to science, and it's somebody who studies profanity. And I just thought it was an interesting topic. Yeah.
Hey.
So he talks about the fact that, in all cultures, there are basically 4 categories about which there's profanity: religion, sex, body function, and groups of other people. He talks about how taboo words change over time. One of the things I found interesting was that there's a word that's worse than the F word in Great Britain. In fact, it's the 4th worst word there.
Right.
And in American interest, it's just a silly word. So you can get in a lot of trouble if you don't know what you're doing. And by the end of it...
Would you tell us what that word is?
You can read the article.
Can we even say that on an international podcast?
Probably not.
It's interesting knowing this, too, because I would have used that term. These people are just,
Beep.
that.
Yeah.
And,
so you just have to read the article. But they also talk about the fact.
Prankers.
That.
Oh, yeah. No. I don't know that word. I do know that.
4-letter words
I know
are what they are because of the kinds of consonants that they begin or end with. And by the end of the article, the writer gets the cognitive scientist to tell about his most favorite word. So you have to read that to see it.
Yeah.
So.
Yeah.
Just an interesting take on language and culture.
Yeah.
My reference text.
Apparently, there's a very bad word in English that is can even be a compliment in Australian English.
Mm-hmm.
So
Yes.
My reference text for all this, Kathy, is the book called The Limerick. I made the mistake once of buying that book and bringing it home, hoping to be able to read this at dinner time to my kids and wife.
Yeah.
I couldn't get past the second line. Right. And eventually, I was hysterical because I could read the poems and my kids are sitting there like, read the rest. And I said, no, I can't read this one. So I put this book up on the top shelf of our bookshelf because we had a sort of a built-in bookshelves that I put together. This book kept migrating from the top shelf to the middle to the bottom, and I'd put it back up to the top. My kids would get out the ladders, and they probably read every one of them, but hysterical stuff.
My pick is a New York Times article called "Radioactive Boars in Fukushima Thwart Residents' Plans to Return Home." And this is just so surrealistic,
Yes.
They've abandoned the area for some time since the disaster in the nuclear plant, and the animals have gone crazy, running around. And it turns out there are lots of boars. They've multiplied extensively. Their numbers are huge, and they're radioactive. They have cesium-137 in them, and they don't know what to do with them. So they want to shoot them, but they don't have enough mass graves. And they said
you can't eat them.
Can't eat them, and they don't have enough people to put them in incinerators. So they don't know what to do. But the boars are not the only problem. There are other animals running around wrecking things. And this is weird... Go ahead.
The boars are going to be a huge... This was a very interesting article, and I've followed a little bit of this, the wild boar issue, the feral swine issue in the U.S.
Yeah.
This is an epically difficult invasive species to deal with, and they are rampant across most of the Southeast.
That's right.
And which is kind of how I got interested in them, because they're a lot of my kinfolk have been concerned about the wild boar population, and they're incredibly difficult to control, even when they're edible, which they are in the U.S.
Right.
And apparently, you have to wipe out something like 40 or 50% of the population per year in order to make any kind of inroads because they just reproduce so fast.
Australia has a similar problem.
Well, with everything.
With boars too. I mean, they have an annual hunting expedition, you might call this, with jeeps and automatic weapons and all kinds of crazy stuff.
Oh, in the southeastern states, North Carolina, South Carolina, all the way down through Texas, they basically opened up hunting.
That's right.
And of course, these are states where a lot of people will do that, and you can go out and
Yes.
gun these things down most times of the year, and they're made of bacon, and they still can't get rid of them.
They've all got trichinella.
Of course they've got trichinella, but these, you barbecue them.
They have deer hunts in New Jersey regularly to
culling.
control the
We call it
population.
culling.
Oh,
but still.
deer at least are a native species.
Yeah.
That's right.
I mean, this situation is surreal, and it's the same thing is happening in Chernobyl.
I was going to say.
All kinds of animals are coming in. But the reason I like it is this idea that when humans abandon something,
exactly,
the animals move in. And they were there first, right?
They're
just coming back.
No, that is a hope.
Well, the advantage in Chernobyl is that they've also got all the way up to the top predators. They have wolves.
True. Yeah. Yep.
And when you have wolves, you don't have to worry about things like swine overpopulation because the wolves will take care of that.
I know someone from our Department of Pathology who goes over to Chernobyl every year and takes samples of tissues and brings them back and does genetic analysis on those. Mm-hmm. Lots of crossovers, lots of weird-looking chromosome patterns.
Well, a lot of animals are thriving despite the high radioactivity levels.
That's the mystery, isn't it?
And in fact, there are fungi growing in the abandoned reactor.
Mm-hmm.
They can send robots in, and they're actually dependent on high levels of radiation to grow. They've become dependent on radiation. In fact, papers have been published by Arturo Casadeval on this. It's really amazing. Really amazing.
Life refuses to take no for an answer.
Do you have anything you want to share with us, Brianne?
This week on Twitter, I found a really cool link to a page called How Herd Immunity Works. Mm-hmm. And it lets you put in different reproductive numbers and different vaccination rates with a slider, but then actually shows you the disease moving through the population and shows you percentages.
That's nice.
And so it's pretty nice to show sort of how a naive population would respond or how a vaccinated population would respond. And I'm definitely hoping to use it in my immunology class this semester.
Cool. Can you send me that link?
Yes.
Put it in the show notes.
Right now.
Yeah, the animated things are cool.
They're visual.
Yeah, and people have problems with herd immunity.
What did you say?
I have a listener pick from Margaret. To bring two of my loves together, here's my self-serving pick of the week. My knitting patterns for socks and fingerless gloves featuring a DNA cable that I designed. The cable is pretty accurate in both major-minor groove ratio and in a number of bases per full twist of the helix. In addition, the sock pattern features a visual pun on a historic style of knitted socks, where a band of pattern runs down the side of the sock and splits so that half runs down the side of the heel flap and half runs down the foot. In my socks, the double-stranded DNA denatures so that one strand runs down the heel and one down the foot, giving the pattern its name denature. The fingerless glove pattern features the DNA denaturing around the thumb and annealing to keep running up the hand. These pieces aren't really bold enough to be perfect for the Science March, but if you are knitters or no knitters, e.g. Dickson's wife when she finishes Dickson's brain hat, I hope you'll enjoy combining your love of science with fiber. You're getting a brain hat?
I am.
These are lovely. They're on Ravelry.
That's great.
Ravelry?
I immediately emailed it to 3 knitters that I know.
My wife will get this tonight.
Ravelry. Yeah, we've had Ravelry on here before.
I like it.
Maybe from the same person, Margaret.
It.
But yeah, there are lots of cool things out there. There are viruses, phages, and knit and all this. But these are cool. And so the fingers are cut off. They're not present. They're not cut off. They're just not present.
Right.
So that you can
They're
do
not
things.
knit in the
There's
I have about 4 or 5 pairs of those.
Those are called mail mitts. They're called mail mitts.
Yeah.
Fishermen use them a lot because the fingertips are still
cool.
available,
like
but
that.
the rest of your hand is not.
Maybe this should be our image of the week here
Mail mitts.
for the episode. Oh, she shows pipetting with those gloves. Yeah, look at that.
Oh, nice.
If you're in the cold room, that's perfect, right?
Absolutely.
Look at that. Very nice.
Right.
All right, so this is from Margaret, who reminded us that orzo It's not rice. Thank you, Mark. You can find TWiV at iTunes, also at microbe.tv/TWiV, or, on your smartphone or tablet. You've got all kinds of programs you can use to play podcasts, and just subscribe for free so you get every episode. Please subscribe. Do it. Go to iTunes or on your phone. Do it now. Right now. Just subscribe. Okay? We'd appreciate it. And, of course, we love getting your questions and comments. TWiV at microbe.tv. Also, consider supporting us. Over to microbe.tv/contribute. We have a number of ways you can do that, including Patreon, PayPal. You can donate your old cars and sailboats. No, we can't do that.
But if you've
I
got
thought you
some
didn't
really
listen
nice
to
ones,
NPR.
Is that what they do?
Exactly.
No, I don't. I see these billboards on the turnpike that said, give us your old boat or car.
right?
There's a really obnoxious ad on television that keeps playing. These little kids dressed up in 60s outfits and black and pink and every time it even starts, I turn it on and go to another, and they want you to Cars for Kids. That's what it's called.
Over on Patreon, if you give a certain amount of money a month, you get rewards like a mug or a book or whatever. But $1 will get your questions to the top of the queue. That's a good reward. That's a good reward. Whatever. You can bet. Dickson Despommier is at livingriver.org and also parasiteswithoutborders.com. Thank you, Dickson.
You're welcome.
Wasn't this better than cooking for 26 medical students?
Just 20, but yes, the answer is yes. Because I'm always the sous chef anyway.
What, you make sushi?
No, sous chef.
I know.
You know,
I get it. Kathy Spindler is at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This is a lot of fun.
Alan Dove is at alandove.com. He's also on Twitter, Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
Our guest today is from Drew University in Madison, New Jersey, Brianne Barker. Thank you so much for joining us.
Thank you so much. This was great.
You have fun? Yeah.
I had a lot of fun.
It's different in person, right?
It's a lot different in person.
We're not so bad after all.
Oh, this is great. Although I feel like I should have been getting pipetting done. I'm not used to listening to you all without a pipette in my hand.
Yes.
You can go home and listen to this then.
That's true.
Are you heading back now?
I will be heading back soon, yes.
Have we mentioned her jewelry on the air?
We didn't. I have a picture of it, though. Tell us about your jewelry.
Yes. I have a necklace that is a T4 bacteriophage that I bought at the Museum of Natural History. I always wear it when I do any virus lectures in class, which means that my students in virology will get it for a whole semester in the fall.
Do you think, because biomimicry is such a common thing nowadays, that the lunar lander model, you got their idea from this? Because they look similar. They do look similar.
That is a good question.
Yeah, when you need legs to land.
I think that was more convergent evolution.
Probably.
Convergent. Okay, fine.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the sponsor of this episode, Blue Apron. And the new music you hear at the beginning of TWiV is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.